In this video, I'm going to introduce the process of spermatogenesis and its endocrine control. Let us first get a little perspective about where we're discussing. This is a testicle, the scrotum, and within it, the testis or the gonad of the male. Note that the testis is made up of tubules called seminiferous tubules. Within each of these tubules, the process of spermatogenesis is going to generate spermatozoans, or sperm, and ultimately those sperm are going to be moved along these tubules and stored in the epididymis. They are stored, they are nurtured, until ultimately they are ejaculated via the vas deferens. So let's zoom in on one of these seminiferous tubules. Here we see a singular tubule in cross-section. Around the perimeter are peritubular myoid cells. MYO should always indicate to us that this is muscular tissue. So this is a muscle layer around the tubule, the seminiferous tubule. And ultimately, this is going to be really important in completing these peristaltic contractions, pushing spermatozoans, or sperm, which are immobile until they are ejaculated, up the seminiferous tubule, and ultimately to the epididymis. Another very important cell within the seminiferous tubules are these purple cells here, which are called Sertoli cells. They're called cystenocytes, they're called sustenacular cells, and they're also called nurse cells. So a lot of different names. Ultimately, I'm going to call them Sertoli cells. These are making up the bulk of the volume of the tubule, forming this perimeter here. Within, or kind of embedded within these larger cystenocytes or Sertoli cells, are spermatogonia. So over here we can see the word spermatogonium for singular, spermatogonia for plural. These are the germ cells of the XY or male individuals. We can see that they are on the basal side, so closer to the outside of this tubule. Now, during the XY individual's development, so from the time he is born until the time he reaches puberty, his immune system, which is, a bunch of cells carried within blood vessels and kind of completing surveillance of all the tissues of his body, those immune system components are learning essentially that these cells, right, these spermatogonia, germ cells of the male, are self cells, right? So these look the same as any other cells in his body, all the somatic cells, because they are genetically identical, right? They have all of his DNA. However, when the XY individual reaches puberty, these cells begin the process of spermatogenesis, and ultimately the sperm that are produced and all of the intermediates along the way are going to be different genetically than these original spermatogonia. And so as far as the immune system is concerned, these sperm and all these intermediates here are not the man's cells. And so they need to be flagged for destruction and ultimately eliminated. Of course, this is not ideal if you are trying to reproduce. And so in the seminiferous tubules, the Sertoli cells form a barrier between this yellow space right here, which is just the interstitial fluid that is exposed to whatever is in the blood, and the lumen of the seminiferous tubule. And so this is where the spermatozoans, the sperm, are going to be. Okay, so ultimately, the immune system via the blood can see, right, they can complete surveillance on these spermatogonia, the germ cells, because they look the same as any other cells in the man's body. However, there is a barrier that is formed by the Sertoli cells, specifically tight junctions between the Sertoli cells, that prevents anything from the blood from actually getting into the lumen of the seminiferous tubule. This is called the blood-testis barrier. And it's very important and it's necessary because the immune system is trained what is self, what is non-self, before the XY individual starts completing the process of spermatogenesis. Okay. Once he reaches puberty and throughout the rest of his life, these germ cells are going to replicate their DNA, so synthesize a copy of DNA, and undergo the process of mitosis. At the end of mitosis, what is produced are two different daughter cells. Each of these daughter cells is genetically identical to the original spermatogonium. All right, what we can see here is a copy of chromosome number 2, for example, which came from his mom, copy of chromosome 2, for example, which came from his dad, okay? And same thing over here in this cell. Okay? Daughter cell A is going to remain a germ cell. So essentially, this original cell just replaced itself with a new one. Okay? The other daughter cell, however, is going to proceed down deeper and deeper through the cystenocyte or through the Sertoli cell into the adluminal compartment, so into this area that is beyond the blood-testis barrier, and it is going to proceed through the process of meiosis. Ultimately, 4 genetically unique haploid cells are produced from each of these daughter B cells from the initial mitotic process of the spermatogonium. Okay, so let's look at how exactly that works. All right, so we're zooming in here. This is another of the seminiferous tubules. Okay, and what we can see is the initial spermatogonium. Right, it is a diploid cell, so 2 copies of all of those 23 chromosomes for a total of 46 chromatids. Okay? Now, this entire process is going to be driven by follicle-stimulating hormone, FSH, which is a gonadotropin hormone released by the anterior pituitary gland. When FSH binds to its receptors on the Sertoli cells, it is going to trigger the spermatogonium to prepare for and undergo mitosis. So what we can see here is, again, the same thing, chromosome 2 from mom, chromosome 2 from dad. FSH triggers this spermatogonium to enter into S phase, so to synthesize a copy of each of these chromosomes, ultimately producing a copy 1 from mom, copy 2 from mom, copy 1 from dad, copy 2 from dad. So essentially 4 copies of chromosome number 2, for example. And this is happening in all of the chromosomes. It's for a total of 92 individual chromatids. Okay. After G2 and all the checkpoints and everything, mitosis is going to proceed, producing daughter cell A, which is essentially another spermatogonium, and daughter cell B. Now, daughter cell B is pushed down through the Sertoli cell, and now we're going to call it a primary spermatocyte. The primary spermatocyte is going to be triggered to begin the process of meiosis by testosterone and ABP. And so before we talk about that, what the heck is going on here? Okay, FSH, follicle-stimulating hormone, binds to its receptors and not only triggers the process of mitosis, but also via a cyclic AMP second messenger system is going to lead to the synthesis of ABP, androgen binding protein. Androgen binding protein binds to an androgen, testosterone. So where did this testosterone come from? Let me introduce another type of cell. Outside the seminiferous tubules are a series of cells called Leydig cells. They're also called interstitial endocrine cells because they are between the seminiferous tubules. They're in their interstitium, and these are really important endocrine cells. Okay, I will call them Leydig cells because L, Leydig cells, binds to LH, luteinizing hormone. So LH, another gonadotropin from the anterior pituitary gland, binds to its receptors on the Leydig cells, inducing a cyclic AMP second messenger system to increase the activity of STAR. STAR is important in beginning the process of steroidogenesis. Remember that this enzyme, or this molecule, is going to bring cholesterol into the mitochondria and initiate steroidogenesis. Ultimately, this is going to produce testosterone. At the very end, testosterone is produced. Now remember that testosterone is not, it is hydrophobic, it is lipophilic, and therefore it is able to just slip over the membrane of the cell. It can go into the blood, right? And also it can slip into these Sertoli cells. And now it would just all diffuse away if not for ABP. So ABP, just like sex steroid binding globulin in the blood, is going to bind onto specifically this androgen and keep its concentration really high within the testis. So testosterone is super high in the testis because of ABP. Now together, this combination here is going to induce meiosis. Okay, so just a very brief summary of meiosis. Once again, you get this duplication event. Chromosome from mom, copy of it. Chromosome from dad, make a copy of it. And unlike in mitosis, there is going to be a process that's called crossing over at this step. So before meiosis 1 really gets underway, some of this DNA is going to mix up. Some from here is going to go over here and here to over here. So ultimately, each individual chromatid is going to be a little bit different. Okay? It's going to have a combination of alleles from mom and from dad. Okay? So meiosis 1 proceeds, and ultimately, these pairs of sister chromatids are not separated. Instead, the entire pair is pulled into one of the daughter cells. So this pair to here, and this cell, or this pair to here. And so at the end of meiosis 1, you still have these pairs of sister chromatids, but this is essentially a copy of this. And so it's called a haploid cell, even though there are still 46 chromatids individually. I also want to point out that these cells are not separated, so there is no cytokinesis. Right? The two daughter cells are still attached to each other. Okay. Meiosis 2 is going to proceed, and essentially this is when this chromatid is pulled from this chromatid into different daughter cells. Okay, so now we finally have 4 genetically unique haploid daughter cells. We call these spermatids, and over the next few days, these spermatids are going to mature via a process called spermatogenesis. Okay, genetically, they're what they're going to be, right? Genetically, they're not changing, but they are going from these, like it's a chain of little cells, it's all attached, to independent independent spermatozoa. Each spermatozoan or sperm has a little acrosome which helps to penetrate the different layers on the outside of an oocyte or an egg. It has a midpiece, which is essentially energy, a lot of mitochondria, and also the main body of the sperm is going to have the DNA, right, this haploid genome. Also, a little flagellum is going to grow or be formed so that ultimately, after ejaculation, these guys are able to swim towards the oocyte or towards the egg. Okay, so again, this is the process of meiosis. It produces 4 genetically unique spermatids. So that is the process of spermatogenesis, and finally spermiogenesis is going to further develop these spermatids into spermatozoans or sperm. And then again, they are going to be pushed up along the seminiferous tubule via these peritubular myoid cells. Okay. They are going to be stored in the epididymis. Okay. And keep in mind that testosterone is really important in maintaining the sperm within the epididymis. Okay. Also, prolactin, a hormone that is generally probably associated more with females because it is pro-lactation, but it is also required to maintain sperm. Ultimately, during an ejaculation, or prior to an ejaculation, the sperm is going to be moved in its testicular fluid up the vas deferens towards the ejaculatory duct. Now, this process is mostly mediated via the autonomic nervous system. However, there are oxytocin receptors lining the vas deferens. In the female reproductive system, oxytocin is really important in muscle contractions, both of the mammary glands as well as of the uterus. And here, it has a role in the male reproductive system as well, specifically to help with these peristaltic contractions of testicular fluid with sperm, again, towards the ejaculatory duct. And now there are a couple other details that I want to mention before I finish this video. FSH not only induces mitosis, not only induces the synthesis of androgen-binding protein, but also the formation of a hormone called inhibin. This inhibin is really important in the negative feedback on the follicle-stimulating hormone releasing gonadotropic cells. Inhibin essentially inhibits future FSH release. So essentially, FSH is driving this entire process of spermatogenesis. And so if there's enough sperm, essentially, FSH is going to be inhibited by inhibin, which will decrease the stimulus of this process in the future. Okay. Also, there is a role of estrogen in this process. Okay. Remember down here in the Leydig cells, LH induced the synthesis of testosterone. Now, not all of that testosterone is going to drive spermatogenesis. Some of it is going to be converted by the enzyme aromatase into estrogen or estradiol, and estrogen is going to be, is going to have a role in the process of spermiogenesis. Okay, so here we can see that estrogen has some kind of a role that, to my knowledge, is not fully understood, but estradiol is going to be important in the maturing of spermatids into spermatozoans. It has been shown that individuals who cannot synthesize aromatase or have aromatase blocked do not have mature sperm. So they can get to the spermatid phase, right? Meiosis proceeds as usual, but they can't go from the spermatids to spermatozoans. And so there's some kind of a positive role of estrogen on this final process. Okay. Also, the spermatogonia themselves synthesize estradiol. Okay, so one of these spermatogonia, or all of these spermatogonia, are also synthesizing a certain amount of estrogen. In fact, about 60% of the estrogen that is synthesized within the testicles is from germ cells, so it's from these spermatogonia. Okay. What this diagram is showing us here is that the spermatogonia release estrogen, and it's essentially acting as an autocrine factor. So it is talking to itself, the cell talking to itself, estrogen flips back around, it's going to bind to a surface estrogen receptor called G protein coupled receptor 30, and in response to estrogen binding to its surface receptor, this is going to lead the spermatogonium to complete apoptosis. Okay? So what this is doing is limiting how many spermatogonia there are, and therefore knocking out this individual cell is going to knock out 4 sperm. So this is kind of antagonistic to what testosterone is doing, but again, estrogen is important in finalizing the sperm themselves. So there must be some kind of an ideal balance of both testosterone and estrogen in order for males to be fertile. Okay. One more note about estrogen, particularly in animals that have seasonal breeding, estrogen is really important in limiting spermatogenesis when the females aren't fertile. Right? So if an animal breeds in the spring, estrogen is going to be downregulated in the spring, and it would be upregulated at other parts of the year, so that you're not just continually making more and more sperm for absolutely no reason. So once again, this is the process of spermatogenesis. It is occurring in the seminiferous tubules of the testis. Okay. It is driven by FSH binding to Sertoli cells and testosterone binding to ABP. Testosterone comes from the Leydig cells in response to LH, okay, and driving meiosis finally to produce 4 genetically unique, independent spermatozoans. This process does not begin until the male reaches puberty, and so unlike in the female, there must be a barrier between the gametes, the sperm, and the blood, which is called the blood-testis barrier, and it's one of the many things that Sertoli cells are responsible for. Thank you so much for watching. I hope you learned a lot about spermatogenesis.